I'm going to get us started. Thank you for joining us on this lovely day at the beautiful Bass Lake Park. Some of us are here in person. We're at Bass Lake in Holly Springs, but several are also joining us online this evening through Zoom because this is a hybrid event. It's very exciting to be able to offer that tonight. Our host for this evening is the South Wake Conservationist chapter, a community wildlife chapter of the North Carolina Wildlife Federation. My name is Lacey Kegley. I'm going to be your moderator this evening. So in the aim of moderation, we're going to aim to have questions throughout. And if you are joining us online, you can plug those questions into the chat function or into the question and answer function of Zoom. And for those of us joining us live, then raise your hand. Monty will run around with a mic and we'll get those questions answered. And without further ado, I'm going to turn it over to Monty Murray, the president of the South Wake Conservationists.
Thanks, Lacey. Okay, so tonight we have one microphone, not two, because one of them's not working. I am Monty Murray. I'm president of the South Wake Conservationists, and we are an all-volunteer organization. We're part of the North Carolina Wildlife Federation serving the Raleigh area. And our mission is to conserve and improve wildlife habitat and wildlife and diversity through conservation projects and public outreach. I'd like you to check out the different ways that you can get involved at the back table over here, those here in the room. There's a lot of good information there and lots of QR codes. If you need help navigating QR codes, see me. It's a good way to get signed up. We do have an interest survey we did at a recent workshop, and if you haven't done that, I encourage you to do it. Let's know what you're interested in doing. We want to get you engaged. I want to start out with some news about our 3-day Raleigh Habitat Steward training, which was completed this past weekend. It was delivered in collaboration with the North Carolina Wildlife Federation, and it's the first in a series that NCWF is going to be holding across the state. So we're pretty excited to be the first one. We had 29 students, and they are certified now and committed to actively working in support of wildlife conservation. And it includes both existing and many new members of the South Wake Conservationists. The training was, I think people agree, pretty intense, long days, but worthwhile, hopefully. And the feedback was pretty enthusiastic, and we had a final rating. We tallied it all up of 4.8. That's out of 5, so that's not too bad. Many are here tonight, and I'd like everybody who was certified this past weekend to please stand up. Congratulations. Also, we have some new folks joining our leadership team. In March is when we do this changing of the guard, and more on that after this presentation. Tonight, we're going to learn more about how the data that's collected in community science projects are used. What do they do with that data? And there'll be examples from research involving birds, plants, amphibians, and more. Our speaker is the NCWF Conservation Coordinator for the Triangle area, who works very closely with our chapter, so we all know her very well. Glad to work with her. She has graduated from Meredith College last year with degrees both in biology and environmental sustainability. She's conducted pathological research on amphibians in Wake County, as well as field research on tropical ecosystems in Costa Rica. That sounds pretty cool. So please welcome Kara Solomon.
Thank you, everyone. Thank you for the warm welcome. Yes, my name is Kara Solomon, and I'll begin by starting out with a little bit more about me. So I was born and raised here in the Triangle area, more specifically in Wake County, and I grew up with love for the outdoors, being outside, and just generally enjoying nature. And as I went through high school, I started to really develop an interest in biology and taking my AP environmental science classes. I really started to enjoy a little bit more about nature, getting outside, being outdoors. And So as I started to look and decide where I wanted to go to college, what was really important to me was having small class sizes, getting to know my professors. And so that kind of led me to Meredith College. I was also making my college decision during the pandemic, so that also had something to do with it. I wanted to be close to home. So here are a couple of pictures of me. As Monty said, I studied abroad in Costa Rica, so that's me on a boat as we were going between locations in Costa Rica. I did research while at Meredith College with amphibians. I'm going to tell you a little bit about that research tonight, along with a couple of other community science projects that I was a part of. So in the bottom corner, it doesn't really show up on the TV, but that was a picture of me out in the field doing some research. I graduated from Meredith College, so that's my graduation picture. And I've always really enjoyed getting outside, hiking. There's so many great places to go in Wayne County, and I love getting out and doing it with my dog Hooper. So yeah, that's a little bit about me. I have 2 degrees from Meredith College. And yeah, as I continued my education, I just fell more in love with our outdoors, especially here in North Carolina, where I'm used to. So a little bit about the presentation that I'm going to be doing tonight. So we'll start off talking about some of my research that I did while at Meredith with amphibians. It's not a community science project. It's an ongoing research project at Meredith. Sorry, the color doesn't show up super well here. But anyway, the point is that it's a gradient. So we'll start out talking about a project that I was heavily involved in. That's not a community science project. And then we'll talk about City Bird, which is a community science project that I was a part of while in college. But it's open to people on college campuses. So it's not one that's super easy to get involved in. And then we'll finish talking about 2 citizen science projects that are here in North Carolina, super easy to get involved in, and projects that I worked on and participated in while in college, and also projects that I use data from. I pull data from for different classes. So we'll start out not community science, and we'll finish with community science projects that are widely open to the public. So this is the very jargony scientific title for the project that I worked on while at Meredith College. I'll explain a little bit more about it as we go on. But basically, my research project was focused on amphibian populations in Wake County, more specifically in Wake County parks. And we were looking at 2 different fungal pathogens or diseases that are spread by fungus that are common in Wake County. So a little bit of background information about the amphibian diseases that I was studying. I was looking at BD and B-Sal, which are the short-term names for Bactridium densitatis and Bactridium salmonidorians. And those are 2 fungal pathogens that infect amphibians. So the first one, BD, is a very common amphibian fungal pathogen. It originated in Southeast Asia and then quickly spread, particularly through the pet trade, and became an invasive pathogen throughout the world. It has caused devastating population loss in amphibians in South America. The golden toad is believed to be one animal that was infected and potentially extinct in part because of that fungal pathogen. So BD mainly infects frogs and toads. Salamanders can be a carrier for it, but it's not super detrimental to their health. B-Sal mainly infects salamanders, and it can be extremely devastating. B-Sal is also native to Southeast Asia, and the populations there, it doesn't affect the amphibians in that native range, but it spread widely, again, believed to be through the pet trade, and has caused widespread population decline in fire salamanders in Western Europe, particularly Germany and Belgium. And so I was part of a task force. You see the acronym here, SNAPS. It's the Student Network for Amphibian Pathogen Surveillance. And I was part of a task force there to determine whether or not B-Sal had made its way into North America. So part of our sampling efforts went to determine whether or not that disease had made its way here. So a little bit about why these pathogens are so devastating. It causes the amphibian skin to slough off or like come off in chunks, which is incredibly detrimental because amphibian skin it's the way that they breathe, it's the way that they regulate their temperature. If you've ever heard of amphibians being called an indicator species or a bioindicator, they are the presence or absence of amphibians can be a good indicator of the health of the ecosystem or a stream. So if you're in a wetland area that doesn't have amphibians, it can be a sign that it's not a super healthy aquatic environment. So their skin is very sensitive. And so this pathogen comes in, it can be spread by water, it can be spread through soil, it can be spread through contact with infected individuals. So it spreads pretty easily in the environment. And again, it causes their skin to slough off, which can be super devastating for their health. There's no known cure for these fungal pathogens, except for kind of a heat treatment. But most of the amphibians that can contract it they don't do super well under heat or dry conditions. They need that moisture and coolness, dampness. So there's a potential that climate change could be beneficial because it could, in theory, take out this disease, but whether or not it would take out the amphibians first is kind of the question. So unfortunately, there's no known cures, at least not that could be easily applied in the field. There are some laboratory studies that show a couple of promising signs, but again, not super common or likely to be effective out in the wild. So those were the two amphibian diseases that I studied while at Meredith. So I'll talk a little bit about the methods that I use while sampling. So part of my research was to apply for the permitting, to apply for funding for the research. So I did all of that. But then once we were ready to go out into the field, we did a combination of testing and going out to different Wake County parks and determining how likely we thought it would be to find some different amphibians, and then we ranked them, and that kind of determined our sites for where we wanted to begin our research. So while we were out in the field, we would go to a Wake County park, we'd make sure everything was sterilized and sanitized and ready to go, and then we'd go looking for amphibians. It was a really fun way to spend a morning or an afternoon. So while we were looking for aquatic salamanders, we would use the dip net, which is up there. It's a canvas net with a mesh lining. You can drag it along the floor. It's a great way to catch a lot of macroinvertebrates in the soil, but it's also really good for catching tadpoles or the aquatic larvae of salamanders. But it's also good for catching frogs. And so we would either use our hands to catch the more land-based terrestrial species, or we would use the dip net for some of the more aquatic species. And then we put them in a sterile critter keeper as we prepared our supplies. And then we would swab them with basically a sterilized Q-tip with a little with a little bit of deionized water. And so we weren't just like sticking a dry Q-tip on them. So we made sure to wet the swabs, and then we would swab them in duplicate back and forth, hitting a couple of places on their bodies that were known locales for carrying this fungal pathogen. So we'd swab their front, their back, the inside of their legs, swabbing all over to increase the likelihood of detecting something if there was fungus on their body, or it could be detected in the environment as well. Like I said, if it was in the water or the soil, the swabs would have caught that as well. So then we put them in little tubes, put it on ice, and froze it. And then once we got back to the lab, we kept it in a -80°C freezer, which is very cold. So we would go out to these different locations, we'd swab all the amphibians we found, and then we would release them right back to where we got them. So as all in all, the process of collecting them, swabbing them, and releasing them back into the environment was probably 5 minutes or less. We tried to make sure to minimize their stress as much as possible, aside goal of this project was also to look at the amphibian stress hormone, which is corticosterone. So part of it was also to determine how stressed these individuals were. So we tried to do this as quickly and less stressful as possible. Other ways of determining infection are much more invasive or require killing the individual in order to determine these things. So all of our field methods were trying to be as non-invasive as possible to decrease their stress and make sure that we could get them back into the environment as quickly as possible. So I mentioned before a couple of the sites that we went to. We visited all of the Wake County parks. Beach Bluffs just opened, so that one's not included. But all of the Wake County parks, we visited them all and kind of determined how likely it would be to find amphibians. So bodies of water with lots of fish aren't great for finding amphibians because fish like to eat amphibian eggs. We steered away from places that had big lakes or ponds with lots of fish. But we did visit all those sites. We also sampled at Meredith College because it's easy. There's a pond that had been rebuilt at Meredith. And so there were lots of salamanders and aquatic frogs, toads, things coming back to the environment. So we sampled at Meredith. We went to a bunch of different Wake County parks, and we also went to the Triangle Land Conservancy property, Swift Creek Bluffs, because that is a place that is known to have really good salamander populations. So those were kind of our sites that we went to. We didn't swab at all of those sites, but we at least visited them to determine whether or not they would be a good site. So for the amphibians that we sampled any amphibian that we could find: frogs, toads, terrestrial and aquatic species of salamanders. But we also had a special project where we swabbed some Neuse River water dogs, which are a larger species of aquatic salamander. They only exist in the water. They have not evolved to be on land. So we did a little bit of research sampling some Neuse River water dogs as well. So again, the primary objective of my research was to contribute data to the SNAPS program and determine whether or not BD and B-cell were present in Wake County. We had seen some preliminary evidence to suggest that BD had been found in Wake County in the past, but it was hard to determine where and what species might have been affected. So we were part of a team trying to figure out where BD was, if it was here, and what species were infected. We know the different types of groups that are most likely to die if they're infected with BD, so that also kind of played into the research as well. So I presented my research at a bunch of different conferences. Meredith College had the Celebrating Student Achievement Day, so I presented that research a couple of times there. I also went to the joint meeting of ichthyology and herpetology, which is an international conference, which was held in Norfolk, Virginia, and we presented our poster there. So got lots of experience talking about my research. So now I'm going to talk a little bit about some of the species that we sampled and where and when. We most commonly sampled at Meredith College because they have some huge bullfrogs and bronze frogs in their pond. So these are two bullfrogs that we swabbed. They're pretty large fellows, which was really awesome. So we did a lot of sampling at Meredith College, found lots of different species, some larval salamanders as well, but they're so teeny tiny, it's hard to show up in photos. So this is an example of a teeny tiny salamander larva. This is the spotted salamander. We found quite a few of these along the American Tobacco Trail. So we swabbed those as well. With individuals that are this teeny tiny, I mean, they're probably about an inch long or smaller. For those types of really teeny tiny individuals, we would pool swabs. So we would swab all of the little individuals that we found in the water that we found them in order to increase the likelihood that we would detect something if it was there. So these guys are really cute. The little feathery things that show up here, these are their gills because they're still in their larval stage. They're still aquatic only, so they only exist in water. So that's why they're so frilly, and they, as you can see, These haven't developed their legs fully yet, so that's why they're still aquatic as well. We also had a lot of luck at Turnip Seed Nature Preserve. It's in the northeastern part of the county. They have these really nice, deep ephemeral pools, which are really good for mating. We found a lot of mating eastern newts here in their aquatic phase while they're mating. So we had a lot of luck spotting salamanders at Turnip Seed Nature Preserve. It's a really beautiful place. We also found some northern cricket frogs and some green tree frogs. And so these guys, they're so pretty. These are the eastern newts, and they're super speckly. And we found them in water, so we kept them in water until we were swabbing. So we briefly took them out of water and then put them right back where we got them. And so these guys were during their mating phase when we discovered them in April of last year. We also went to Crowder County Park. We had a lot of luck with toads at Crowder, but we also found this southern two-line salamander and some northern cricket frogs as well. So this is the main pond at Crowder. If you've ever been, this is the main pond. So we were setting up and doing some of our sampling then. We didn't have a table or anything, which is why we're squatting on the ground, looking through all of our supplies, trying to find everything. But like I said, we found a lot of toads. These are teeny tiny little toadlets. They're probably about, I don't know, less than an inch big. And we found a bunch of them. So we were going around with little nets trying to catch as many as we could. So this is another example of animals where we pooled swabs in order to increase the likelihood that we would detect something. So we found a bunch of little toadlets. As you can see, like this is what a little Q-tip basically looks like, so you can tell for scale how teeny tiny they are. So we had a lot of luck with little American toadlets. They're very hard to determine whether they're a Fowler's toad or American toad when they're this tiny, but my professor was there and called for backup from the Herp Society for identification. So as I mentioned, we also sampled at Swift Creek Bluffs, which is not a Wake County Park property. It's a Triangle Land Conservancy property. We got really lucky. They had been doing some construction at the park, and so they had left over some boards, like little 2x4s that they had left in the park, which one way that you can look for amphibians, particularly the terrestrial species, is by putting down cover boards, which basically just gives them a place to hide. It's damp, it's cool, it's dark, which is what they like. So we got really lucky that there had been some leftover what basically worked as a cover board. So we flipped them over and 3 of these little guys scurried out and we were able to catch them and give them a quick swab. They're super cute, teeny tiny little legs, but really big bug eyes. So we had some luck at Swift Creek Bluffs as well. We went back to Turnip Seed Nature Preserve because of how much luck we had had. So this is me with a couple of the newts that we swabbed. We tried a different vernal pool for this specific visit, and we had a lot of luck there as well. So this is a salamander egg mass. If you've never seen one of these before, salamanders, particularly the eastern newts that we were finding, lay their eggs in these vernal pools. And so there are these teeny tiny little green, basically little marbles that contain these perfectly preserved, developing little salamanders. So that's what's in my hand here. And then we also have the whole egg mass, which contains quite a few of these little marbles, and they're surrounded in this protective jelly, which is clear, and that's what's in my hand that I'm jiggling around. So we just scooped it right out of the water, and when the mother lays the eggs, she surrounds it with that jelly, but as it absorbs water, once it's deposited in the water, it grows and swells. So that's why it's this big and the salamanders are so small. So yeah, that was a really cool find. I had never seen an egg mass like this before, and they're so teeny tiny. They were really cute. So we didn't swab the egg masses. There wasn't really a point, but we did find a couple of adults, and so we swabbed those while we were there. And then, like I mentioned, we also did a little bit of research with the Neuse River water dog. This was on a property that was on the Johnston County border with Wake County. And we found a couple of Neuse River water dogs there. These are a couple of smaller individuals, but typically they can grow up to 11 inches. They're an endangered species. They're only found in the Neuse and Tar-Pamlico River basins. And so here's an example. They're aquatic species only. They have those gills. They like to live in riverbeds, moving water, which helps their gills absorb oxygen that way. And so we found a couple of those and we swabbed those as well. So as far as the results from my research, this is an ongoing project. They're still continuing to sample. It's a little bit up in the air now, but they're still continuing this research. This was always a project that was going to live on beyond me. But lucky for us, the preliminary results we sent in are swabs from Swift Creek Bluffs. We also sent in our swabs from Blue Jay Point. They have a big pond by their visitor center, which was just a breeding ground for a bunch of newts and tadpoles as well. But because SNAPS mainly focuses on B. sal, which is the fungal pathogen for salamanders, we focused on at least beginning sending our results to be tested that were mainly just for salamanders. But we did test for both BD and B. sal on those organisms. So we sent in our 3 swab, or it would have been 6, but we sent in our swabs from the 3-line salamanders at Swift Creek Bluffs, and we sent in our swabs from the 13 eastern newts that we got at Blue Jay Point County Park. And we also sent in control swabs to make sure that all of our things hadn't been contaminated, just as a double checking measure. And we sent in all of those and everything came back negative. There was no detection of BD, there was no detection of B. sal, which is really good news. Unfortunately, we sent in some swabs for the Neuse River water dog, and 2 of those came back positive for BD at very, very low levels, just barely detectable. So it probably wouldn't have started to show signs on the individuals. We were recording biometric data, their size, looking for any signs of BD. And so all of the amphibians that we sampled didn't have any signs of infection. But they did test positive for very low levels of BD. Again, there's really nothing that we can do about it except use this to monitor. But yeah, so that was a little bit about what I did while I was at Meredith. That was my main research project that I worked on for a couple of years. But I also did some community science projects while I was at Meredith for various classes while I was there. So I'm going to start talking about some of those now, beginning with City Bird, which again is only a citizen science project that is taking place on college campuses, but it is a community science project that anybody on those campuses can take part in. So City Bird is a community science project that monitors bird window collisions. And so it's only in the Triangle area so far. They have set up surveys at NC State, UNC Chapel Hill, and Meredith College. And their hope is to kind of determine what buildings are at the highest risk for collisions with birds. And typically that's the buildings that have the most glass on them. And then hopefully that can be used to make decisions to make these buildings more bird friendly, more bird safe. So this picture at the bottom is an example of a bus station on NC State's campus. They installed bird friendly glass. It's really a film that they put on the inside of this glass structure so that it's more easily seen by birds. Because with normal glass, a lot of times they just don't know it's there, and so they'll run into it. And so this particular version of bird-friendly glass has a bunch of rows of little white dots, which is something that they can perceive and prevents collisions. So they were also looking to determine what species are most vulnerable for colliding with windows. That was another part of the project. So I participated in a couple of surveys at Meredith College. These are the 3 buildings on campus that they've selected for monitoring. The library, the student center, and the science and math building. These are the 3 buildings that have the most glass on them. Most of the buildings on Meredith's campus are older buildings. They've been around for a long time, so they don't have big, huge glass windows, the same way that UNC Chapel Hill or NC State, which are often newer buildings. So these are a little bit different. So because I was a biology major, I mainly did sampling and surveys around the science and math building. Outlined in green is each building. So when you sign up to do a survey, we picked a time slot, a day, and a building, and then we'd walk around the whole perimeter of the building and monitor to see if there were any birds, either dead or injured, that had potentially run into one of the windows. So I sampled and surveyed the science and math building a couple of times. Luckily, I didn't find any dead birds. But in the 2 years that this community science project was taking place, there were 21 observers that were involved in the project and a total of 35 collisions that were reported. This resulted in 33 fatalities and 2 birds that did survive, but were injured. As you can see, Meredith College had very few compared to UNC Chapel Hill and fewer than NC State University. Again, this is in large part because those buildings are newer, they have more glass, they're more likely to have collisions. So the way that the surveys worked, again, we signed up for a time slot, but if people did find a dead bird that they thought had been injured due to a window collision, then they could log those observations as well. So it did count for animals that, you know, somebody walked up, was going to class and found a dead bird, they could record those as well. So again, much fewer than NC State and UNC Chapel Hill, but still 35 total collisions were reported. So this is the science and math building there in the center. There's that big glass atrium, which was probably the top contender for bird collisions with windows. So hopefully this was somebody's research project as well to increase awareness and really dive into the data on this project. So hopefully there will be measures and steps that can be taken to make this atrium a little bit more bird friendly by installing those stickers that makes it easier for birds to detect the windows there. So these are the 5 species that were most common for window collisions at Meredith College: the American robin, the ruby-throated hummingbird, the mourning dove, the common yellow throat, pardon me, and the black-throated blue warbler. Those were the 5 most common birds that were found to be colliding with windows. So now we're going to move into the section of community science projects that I participated in, that I use data from, and that you can be a part of. So this first one is Nature's Notebook. It is part of the National Phenology Network. Phenology is the study of seasonal changes. It can be used as an indicator of climate change. So, for example, if any of you have read Doug Tallamy's Nature's Best Hope, he talks a lot about some of these indicators of climate change in phenology. So, for example, there can be a mismatch between when one stage happens in another. So, you know, if caterpillars depend on trees for leaves, for food, and birds depend on caterpillars to feed their young, there's that relationship and cycle that takes place. So if trees are putting out their leaves earlier than they had been because of climate change. The temperatures are warmer if they put out their leaves earlier, and the caterpillars are still going based on the time of day. So, you know, it might be warmer, but the time of day, the daylight hours, the photo period hasn't changed. Then there can be that mismatch. So you know, if one is emerging before the other, then, you know, then birds don't have anything to feed their young, that kind of thing. So phenology, particularly phenological mismatches, was something that was being studied with this citizen science project. It's mainly for plants, but it started, I think, in 2013, and since then there have been over 30,000 observers, pardon me, and over 400 million observations recorded, and at over 21,000 different sites. So this phenology network database, when you log observations in Nature's Notebook, they are put into the National Phenology Network database, and that data is readily available. That data has been cited in hundreds of peer-reviewed papers. You can look it up on their website. It's really easy to find. You can look through some of the different papers that have used data from this network. It's pretty neat. So I participated in this project at 2 different sites. The first one was at Meredith College, and the second one was at my house. So this is an example of what an observation log looks like for this citizen science project. When you first have to select a site, so Meredith College has a site, and they have a bunch of different ones actually, and so I had to go in and select my site. And then this is an example of some of the questions you have to answer. It covers all kinds of different seasons, so spring, fall, summer, everything. So you have to record yes or no for all of these questions, like, do you see any breaking leaf buds? Do you see any flowers? Do you see any fruits? Do you see any leaves that are starting to change color? Those kinds of things. If the answer is yes, then you have to quantify it. So there's ranges from 0 to 100 and all the way up to 10,000 or more. So for example, do you see any breaking leaf buds? Yes, I can see them here. Look up to the tree canopy. How many breaking leaf buds do you see? So you have to go through and answer all of those questions, but it's a really cool way for you to get to know some of the plants in your yard and notice some of these changes as they're occurring. So the site that I had at Meredith College, I monitored 5 different pin oak trees, 2 big white oak trees, 1 post oak, and 1 flowering dogwood. And so every week I would go out, I had that checklist, and I had to go out and record how many different leaves I saw breaking, yes, no, pollen, all of that good stuff. So I started, it was probably late January, and we went all the way through May. So got to see it from a dormant tree all the way through to fully leafed out. So that was pretty cool. So as part of the project that I did with my site, we had to do a research presentation with some of the data. So we had to come up with a research question and then report on it, get the data, pull the data from the database, analyze the data, and then present on our findings. So because we had a couple of white oaks in our plot, and there's some really good information about the accumulated precipitation throughout the southeast. So we decided to look at the association between accumulated precipitation and the timing of first day of leaf bud. So we looked at white oak trees that had data from them across the southeast, so across different states between 2011 and 2022. And so we compared that with the accumulated precipitation, which came from another source, but it was all compiled together. And so it's really easy and really fun to play around with this data if you're ever interested. The National Phenology Network database is really pretty easy to use. So we made a couple of graphs. I hadn't taken any statistics or data analysis class, so I'm sorry if it's not super amazing, but this was kind of the general trends that we observed in the data from 2011. Actually, the data went through 2021. And so this was the distribution of leaf budburst in white oaks, based on the day of the year, kind of dips and then goes back up. So it's roughly around day 100. So the 100th day of the year is roughly when the white oaks would start to leaf out. So yeah, that was some of the data that we played with. And then we also mapped the accumulated precipitation with the mean first day of leaf bud burst, and our results suggested that there really wasn't an association. Accumulated precipitation isn't really what's making white oaks leaf out. There must be some other contributing factor. So statistically, they were not associated. So I pulled data from the National Phenology Network database. I created a research project and we played with the data. And so that was one way that we practiced our data analysis and research skills using that database. It was really fun and easy to use, but it was also a great way to learn about some of the plants. All of the plants in Nature's Notebook, when you select your area, it's only native plants. So they're not interested in tracking these things for some of the invasive species or things like that. It's really only for native plants, which was pretty cool. I got to learn about native plants in our area and also learn how to observe when the leaves are budding. What does pollen look like? What does the fruit of this tree look like versus the fruit of this tree? So it was a good introduction into an upper level like plant biology class as well. So I really enjoyed that. So the final citizen science, community science project that I'm going to be talking about is Natural North Carolina. It's a project on iNaturalist, and it's one that I contributed to while at Meredith. I mainly used it for bioblitzes. So if you don't know what a bioblitz is, it's basically when you set aside a certain amount of time and you go out and you document as many different species of plant, animal, fungus, anything, when you go out and document all of that. So I set aside a couple of times throughout one semester from about January to April. I set aside about an hour a week, and I would go to different places, different parks or greenway trails in my area, and I would do a bioblitz. I would document everything that I could find in that area. But to tell you a little bit more about Natural North Carolina as a project, it's a statewide iNaturalist project. Their hope is to kind of create an atlas of the biodiversity of flora and fauna in our state. So the way that you can participate in it is if you go into iNaturalist, you select this project, you can add it to your profile, and then when you go into the app, when you're making an observation, you can select this project and turn it on every time you want to make an observation. So for example, this is an observation that I made at the Habitat Stewards training. I found a really cool rust fungus on a juniper tree. And so at the very end of your log, after you put in the location, the date, the time, all of that stuff, at the very bottom, it says projects. So if you click on projects, after you've added it, you can turn it on. So you turn on the Natural North Carolina project and your observation will be put into this project. And so I made sure to do that for all of the observations while I was doing those bioblitzes. But as far as the overall project, they started this project in 2013, and since then they've had over 114,000 observations, over 8,600 different species have been documented, and over 1,800 different observers have been involved in the project, which is pretty cool. So these are a couple of the different species that I took photos of. Another thing about iNaturalist, if you don't know, you can also take pictures of different signs of animals, or if you've heard an animal, you can record that as an observation as well. So a lot of the pictures that I have are from things that don't move. So took a lot of pictures of plants, a lot of pictures of insects that were doing something, or, slower moving animals, but you can also log observations for different bird species or mammals, whatever. But often the pictures aren't super great for those because they're moving or trying to get away. But so things like plants, Japanese stoat grass, there was a lot of that I observed. A couple cool different mosses, lyre-leaf sage along the Greenway Trail, five-line skink, bronze cutworm moth, and then some cool different fungus, which doesn't have a easy common name, only the scientific name. So I really enjoy getting involved in this project. I've used it since. It doesn't have to be for a BioBlitz. Anything that you observe in North Carolina can be counted in this project, but for the purpose of the academic research that I was doing, I needed to have a BioBlitz be part of it. But it was really cool. I got to go out to a bunch of different greenway trails near my house and explore, observe, which is something that I've always loved doing. And getting other people involved. I would try to recruit people to come with me and we could go out and see what we could find. It was always super fun. So again, this was part of a project that I was doing for class. So we had to record and download data from this iNaturalist project. For the purpose of the class, I was looking at the observations in Wake Forest specifically. So I was charting the number of observations in this project in Wake Forest over time. So as you can see, it started out in 2013. There were less than 5 different observers that participated in the project. It slowly started to increase, but we saw a real spike in 2020. I wonder why. So I thought that was a pretty cool piece of data that we saw about the overall trend in observations in Wake Forest over time. And of course, I should also mention that I took out all of my observations. So this data is, there's a lot more observations, but the only one observer. So in order not to skew the data for the research, I took my own observations out because that was a lot of the observations in Wake Forest. Another overall trend from the observations in Wake County, or sorry, in Wake Forest, were that the most observed species in this project were plants and animals. So this is the taxonomic categorization breakdown of the different observations in Wake County. So the big orange slice, over half of the observations were plants. 21% of the observations in Wake Forest were insects, and then everything else was 5% or less of the pie. So it was a lot of, most of the observations are plants and insects, again, because those are easier to take pictures of and to identify. And then this is a table that I made as part of the project, comparing the total number of observers, the total number of species, and the total number of observations throughout the iNaturalist project versus the breakdown just in Wake Forest. And then, of course, my data separately included as well. So in a couple of months, I made 64 observations of 59 different species. But of course, all of this data is outdated by now. But that's an example of what I did with the data that's readily accessible in the iNaturalist project. Downloaded it, played with it, and was able to kind of get these overall trends in the observations that we saw in this project. So again, just to recap some of the community science projects that you can be a part of, Nature's Notebook is one. The data in that, again, is really cool, especially over time. It's really interesting to look at these different trends in plants over time. So this is something that I have kept up with, not so much at Meredith College, but in my yard, observing and noticing the different timing in when leaves emerge, when flowers emerge, when fruits emerge, the different timing of year. That's a really interesting thing that you can look at over time. So you can join Nature's Notebook by creating a site in your yard, selecting the different native plants that are in your yard and observing those over time, or you can join projects that they already have set up. For example, Prairie Ridge Eco Station has a project that you can join and make observations through that. So Nature's Notebook is really cool. I don't think many people know about that one. It's a really cool community science project. And again, the data for that is really fun to play with and to look at. Another project that you can be a part of is, again, the Natural North Carolina Project on iNaturalist. It's just like iNaturalist, but it's a specific project that you can contribute your data to. All of this goes through the Museum of Natural Sciences here in Raleigh. Their scientists look at this data and use this. And it originally kind of came about through NSF funding. But it's a great way to look at the different flora and fauna that are here in North Carolina. And this is another one that I continue to make observations in. As you saw, why not? I mean, it's super easy, and it's a really interesting way to be a part of the different flora and fauna that are here in North Carolina. And who knows, you might find something that no one ever knew before. So yeah, those are a couple of the community science projects that I was a part of. A little bit about some of the research that I did while I was at Meredith. This is a harlequin frog that I saw while I was in Costa Rica. And I just love that it looks like it's like, hey, what are you doing? So yeah, that's my questions picture. I will take any questions now.
You need to use the microphone so that the people on Zoom can. Go ahead, question. Raise your hands again. John.
When you were in South America, it was Central America, right?
Costa Rica.
Yeah. Did you ever, or did you hear, like possibly when it came to that disease with the amphibians? Was there ever any experiences with the poison arrow frogs?
Poison arrow frog?
Poison arrow, they have kind of a poison skin. Just wondering if you had any observations or experiences.
We used to have 2 mics, fortunately.
Yes, I did. I saw a couple of different poison dart frogs while I was in Costa Rica. One they call the strawberry frog. They're all really small. I did not realize how small poison dart frogs are. But the strawberry poison dart frog is less than an inch. It has the hind legs are kind of a blue color, and so other people call them the blue jeans frog or the denim jeans frog because it looks like they're wearing jeans. And then they also have like a pinky strawberry color to them. We also saw black and white poison dart frogs and I believe one other species, but we did see them. I don't know that they are super at risk for any of the amphibian fungal pathogens, but we did see quite a few of them. Yes.
We have a question online.
We do. Jessica asks, going back to the City Bird Project, was the aim of that project to know if the dots on windows were an effective way to mitigate a bird collision, or was the aim of the project to kind of measure what the impact was to birds that were injured and killed?
Great question. So the main question was to determine whether or not, well, actually, I should mention the Science and Math building and all of the buildings on Meredith College campus don't currently have anything for bird collisions to prevent them. So it was a way to kind of see what buildings, if we're not just putting this on every building for financial reasons, what buildings should we target for these bird-friendly window treatments? What birds are most likely to collide with windows? What specific species are most likely to collide with windows? And then also specifically what buildings should we target for these bird-friendly treatments? So it's kind of a way to determine, what kind of treatment we should put on buildings. There weren't treatments on most of the buildings currently. So the goal was not to determine different methods and which one was most effective, because there was no treatment on those windows.
A lot more hands earlier.
Many years ago, I had the opportunity to do a review of the School for Field Studies in Costa Rica. Were you involved in one of those academies they have in Costa Rica, or was this a study abroad from Meredith College?
No, it was a study abroad program just through Meredith College. So we stayed at different field stations. But we had a local guide that we worked with. But we went with Meredith College faculty and Meredith College students. So it was just a program from Meredith.
So your data showed that the Newt River, salamander, river dog,
News River Water Dog.
Neuse River water dog, a couple of them showed, signs of the BD. Is there anything going forward, or is there gonna be continuing monitoring? Is it something to be concerned about for the rest of the amphibian life?
Yes, this is a project that is being continued through NC State. So they will continue to monitor to determine if it's spread, those kinds of things. So it's an ongoing project to determine and examine whether our findings, which again, super small sample size, to determine if there's a risk of spreading this on to different species of amphibians. Or, is this a fluke? Were their samples contaminated? Those kinds of things. So it is a question and a research project that is being continued through NC State. Meredith may take part in it from time to time, but there is continued monitoring to determine, do we need to be concerned? And if so, where should we target our sampling, and, is it early enough that we can kind of quarantine or, kind of work from there to determine next steps.
Has any of this data or the information you collected gonna be used possibly by people who worked on this for, say, graduate studies or toward an advanced degree?
This data will be continued, but not so much for a graduate degree. It's just general knowledge and data. Hopefully at some point it could be published in a peer-reviewed paper, but not so much for graduate work. Depending on how funding things go and if there is interest, it could potentially be used that way in the future. But at least for right now, this is a project that my faculty mentor was really passionate about. And so it will continue on with the undergraduate students, and then hopefully be published someday in a peer-reviewed journal article of some kind.
Can this fungus affect any amphibians or just the small group that you were showing?
BD can infect any amphibian, and both BD and BSAL can be carried by any amphibian. Whether or not it harms them is dependent on the species, but it is something that can be carried, passed on by any amphibian. So it could infect any group, whether that's frogs, toads, salamanders. But for BD, again, it's most likely to infect frogs, toads, even a little bit less. And then for B-Sal, of course, it mainly infects salamanders, but it could be carried or passed on through any species of amphibian. Again, there are specific species and categories within the amphibian community, excuse me. And those can be, those are at a higher risk. So for example, the species and related amphibians that are in that fire salamander category, so the related salamanders in that category, those would be more likely to be infected and have that infection be lethal. So there's certainly association to it. Some of them are more likely to die, but it's not just that any amphibian is going to die from it. It is more species dependent than that, but it can be carried by most of the amphibian.
Is anyone studying the Asian varieties of salamanders and things and to find out what in their immune system makes them survive this?
Yes, and there's actually a lot of interesting research on the amphibian skin microbiome. So all of the different bacteria and fungi and all the things that live on salamander skin. So there's some interesting research being done looking at the like skin microbiome of different species and whether or not you could kind of create the equivalent of a vaccine or inoculation with some of those beneficial like antimicrobial properties that are found in that skin microbiome, whether you could use that to inoculate species that are infected or that are at risk of infection. So there's some interesting research being done there, looking at the different like antimicrobial properties on some species, particularly the ones that you mentioned that can be found in Asia that are kind of immune to this fungal pathogen. So yes, there's some interesting research being done. There's been a couple of studies that have shown promise, but again, the like, how do we put that into the environment and really spread it widely is kind of the big question there.
And I think you kind of answered one of these questions that we have online, whether this is a fungus or bacteria for BD and B-Sal. Another question is, can birds or fish die from eating an infected frog or salamander?
That's a great question. And yes, both BD and B-Sal are fungus. They're both fungal pathogens, so they're both the type of fungus. And as far as the like effects up the trophic levels, we haven't really seen that. It doesn't appear to infect different types of categories of animals like mammals or birds the same way that it infects amphibians. So more research would probably need to be done about that, but I don't think there's any evidence so far to support that. I don't think that it affects birds that eat an infected amphibian. Doesn't seem to yet.
Two questions really. One, you said these swabs were sent off somewhere, knowing what's happening with federal funding right now. Was that a federal lab, a state lab, or a private or university lab? The second question is, you mentioned when you started out that your professor was also interested in the cortisol levels for stress. Did they do that? Can you get that from a swab on the skin? Most critters, you have to have a blood sample or a urine sample. They've even measured it for wolves in the urine and snow and so forth. So anyway, those are the two questions.
Okay, so for the first question, we stored a lot of the samples in our lab, so a lot of the swabs are still in the freezer for the ones that we didn't go ahead and send away, but they were sent away to a federal lab. I believe it was in Washington, DC. So there is potential, issues there. But we do still have a lot of the swabs at Meredith. So if there's a time in the future, we could send those away. And then as far as the corticosterum goes, there are a couple of early studies that say that corticosteroid levels can be determined from a dermal swab. Oftentimes you're right, it is with a blood assay or a urine sample. And so again, we were trying to do it as non-invasive as possible. So we were part of an effort to determine how effective it was to measure corticosterone from a dermal swab. I don't think any of those swabs were sent away for early assays yet. But yes, we were hoping that our research and our swabs would determine whether or not that's an effective method. Early results were saying yes, but yeah, it also could pick up the corticosterone from the environment in the water, those kinds of things to determine the overall stress level of amphibians in the Wake County parks and things like that. So yeah, I think that's the question.
We have time for one more question if there's another question.
Do you know if that is the fungal disease, if that's found in any of the other states? Or I was thinking, maybe Florida might have a higher risk since in the Gulf tropical waters there.
Thank you. Yes, so B. sal has not been found in any other states. Again, where we're trying to determine whether it had made its way to North America. I don't think it has been determined or been detected in North America yet, but BD definitely has. It is present in other states. Yeah, it is. I'd have to look it up to determine what states. But yes, especially states that have prime conditions where it's damp or cool. Yeah, that's where BD is most likely to spread.